Today I'd like to talk to you about the idea of using DNA sequence data, also known as genomic data, from newborn screening throughout the life cycle.
But first, let's talk about what DNA is. DNA contains the instructions that tell our bodies how to grow and develop. Each person has a unique DNA sequence that affects many things about them, such as their eye and hair color, as well as their chance of developing some diseases. DNA is made up of a sequence of letters, A's, T's, C's, and G's. So what is genomic sequencing?
Genomic sequencing is a technique that is used to read DNA. It finds the order of the letters of a person's DNA one by one. The DNA sequence can be referred to as genomic data. There are a lot of letters to be read, which means that genomic sequencing can produce a lot of data. Once the DNA sequence is obtained, the genomic data can be used by doctors and scientists to help identify the causes of genetic diseases in patients. Doctors and scientists can look for changes in the DNA called variants that are different between people with diseases and people who are healthy. These changes might be very small, like spelling mistakes, or bigger, where large runs of letters are missing. Now let's talk about what newborn screening in Australia looks like.
Newborn screening is a government-funded public health initiative that aims to detect rare and serious childhood onset conditions in babies before they start developing symptoms. Earlier detection of these babies means treatments can be started earlier, which leads to better health outcomes. In Australia, newborn screening programs screen for approximately 25 conditions, although this varies a bit depending on which state or territory you live in. Newborn screening is often referred to as the heel prick test because that is how drops of the baby's blood are collected in the first couple of days after they are born. A nurse or midwife normally does this, either in the hospital or at home. The nurse or midwife also gets consent from the parents to take the blood sample. The samples are tested by a laboratory. Abnormal biochemical levels in the blood indicate that the baby has the early signs of one of the serious but treatable conditions being tested for. Each condition has its own biochemical that is tested. If an abnormal newborn screening result is found, parents will be notified by a health professional and further tests will be done to confirm the results.
As you know, we are running a research project to see whether it is possible to use genomics in newborn screening. Let's recap why we might want to do so.
Because genomics can test many genes at the same time, using it in newborn screening would mean that we could test for more conditions all in one go. This means more babies will be detected with conditions earlier so they can start treatment sooner.
When we look at data in the newborn screening project, we only look at a set list of genes that cause serious but treatable conditions that begin in the first couple of years of life. We deliberately don't look at genes that cause other conditions that don't fit this criteria. It is important to remember that we might have variants in our DNA from birth that could cause genetic diseases, but we may not develop symptoms straight away. For some genetic diseases, we may not develop symptoms until we're a few years old. For others, we may not show signs of the genetic condition until we're in adolescence, early adulthood, or even much older, if at all. This means that looking at our genomic data can predict what kinds of conditions we might get in the future. Because our DNA doesn't change throughout our lifetime, we can also think about whether we should be re-looking at the data at other times during our lifespan. We want to know what you think we should be looking for and at what ages.
Thanks for listening, and we look forward to seeing you in the focus group.